In this tutorial, you will learn how to use NetGestalt's network analysis and node link visualization tools to view your data of interest within the larger context of a biological network. Let's start with the module enrichment tool. The tool is available for single binary tracks and single continuous tracks. Select a track and the module enrichment tool will search all of the gene modules that are statistically enriched with the track's genes. For single binary tracks, a Fisher's exact test is used to evaluate whether the genes in the track are enriched in any of the network modules. For single continuous tracks, genes are ranked based on their continuous values in the track. For each network module, the Kolmogorov-Smirnov test is used to evaluate whether the genes in the module are enriched in either the top or bottom of the ranked list. Let's take a look at an example. Here we have a single continuous track and a single binary track. You can find the network analysis tools under the track title. Let's analyze the single binary track. Select the module enrichment tool under network analysis and click Go. The enriched network modules panel shows the enriched modules found. You can sort by name, p-value, or the enrichment ratio. You can add an enriched module as a new single binary track by clicking on a module name, inputting a new track name, and clicking Add. You can add all of the enriched modules as a new composite binary track using the Add All Enriched Modules option. You can do the same module enrichment analysis for single continuous tracks. But as you can see, when you add all enriched modules into a new composite binary track, a new composite binary track is generated where the enriched modules are colored blue or red based on the sign of the value of the Kolmogorov-Smirnov test statistic. Let's zoom in for a closer look. Notice how some of the genes are either light or dark colored. For the red enriched module derived from the single binary track, the genes are dark red if they are present in the original single binary track and light red otherwise. For the blue enriched module derived from a single continuous track, the genes are dark blue only if those genes contributed to the enrichment signal, and light blue otherwise. Next, let's look at the network expansion and gene prioritization tools, both of which are available for single binary tracks only. When you use these tools, the genes in the track are treated as seeds. When the network expansion option is chosen, a Fisher's exact test is used to find the non-seed genes found to be enriched with your seed genes as network neighbors. Conversely, when the gene prioritization option is chosen, seed genes that are enriched with other seed gene neighbors are identified. For example, if we wanted to perform the network expansion tool on the track of significantly differentially expressed genes, we would select the network expansion option under the network analysis window, then select a false discovery rate, and then input a name for our new track. Notice in this new track how the seed genes present in your single binary track are colored red, while the non-seed genes are blue. Next, let's visualize this collection of seed and non-seed genes with a node link graph. Node link graphs are available for any single binary or single continuous track. There are two options for node link graphs. The first option is to view only the genes present in a given track. Select the node link graph present nodes option. A node link graph showing the network of only those genes in the selected track will be shown. In this case, the nodes are colored red and blue for seed and non-seed genes, matching the coloring in the track. You can rearrange the nodes and export the figures. The node link graph visible range option will show all the network genes currently visible, whether or not the genes are present in the track. Genes present in the track will be colored in. This feature is available only when 500 genes or less are currently visible. Next, let's look at the gene set enrichment option. Similar to the enriched network module analysis tools, the gene set enrichment option allows you to perform a Fisher's exact test or Kolmogorov-Smirnov test on pathway databases or gene ontology terms for pathways or terms that are found to be significantly enriched with the genes in your track. Let's search this binary track for KEGG pathways enriched in these genes. The results are shown in the panel on the left. Now, let's add the cytokine-cytokine receptor interaction pathway. The new track contains the intersection of the genes in your selected track and the genes in the pathway. Finally, Let's zoom out to view all of the genes in the intersection of the enriched pathway. If you want to compare this intersection with all of the genes in the cytokine-cytokine receptor pathway, you can select that path as a system track as described in tutorial 2. This concludes tutorial 5.